Hello, my name is Emil Schmiedl and I'm an undergraduate student supervised by Lara Burchardt at the Humboldt University of Berlin. In this short talk, I want to present a novel method for detecting rhythmic regularities within inter-onset interval ratios of animal vocalizations and possibly other biological sequences. Rhythm is an important aspect of biological communication and behavior across species. There is a growing interest to better understand how biological sequences are temporarily organized. For an instance, recently many studies investigated how the intervals between single sounds in animal signals relate to each other. Simple ratios like 1:1, 2:1, or 2:3 between these inter-onset intervals, short IOI, are especially interesting, since they have been linked to perceptual salience and predictability and are also a prevalent aspect of Western human music. One example for this would be the song We Will Rock You by Queen, where we find the ratios of 3:2. In the much less constrained animal vocalizations, current approaches to investigate those ratios face two problems. Firstly, they only look at the neighboring IOI instead of the whole signal. This can overlook global temporal patterns across the entirety of the sequence. Secondly, some implement arbitrary thresholds that determine whether a given ratio qualifies as close to an integer ratio or not. To overcome these limitations, we propose a new approach. We score all IOI ratios in a sequence for closeness to a small integer relationship on a variable continuous scale. This function assigns scores of rhythmic proximity between 0 and 1 to each pair of IOIs as shown in the bottom right plot. The peaks around the small integer ratios are flexible and depend on the parameters of the function. This way, we can combine isochronous, heterochronous, and complex structures in one go. Instead of deciding if the ratios are met by fixed thresholds, we then compute a set of random sequences with similar properties to the original from an exponential IOI distribution. After that, we can test whether the ratios found in our animal sequence score closer to integer ratios than we see in its random counterparts. By incorporating statistical inference into our analysis, our results are much more data-driven. With rhythmic proximity, we can ask the question whether a recording shows statistically better results than similar sequences with random structure. We tested our approach with various testing sequences, including jitter, isochrony, and heterochrony. Afterwards, we applied the concept to animal data from bed pups, fish, and whale coders. Another benefit is that we can better visualize the relationships found in our recordings. Such recurrence plots, for example, reveal pattern in sequences through coloring by integer ratio, while the intensity equals the rhythmic proximity score of this IOI ratio. To summarize, we developed a novel approach on testing rhythmicity in animal vocalizations, based on a flexible function to score rhythmic proximity and comparison to data-driven null sequences. The method can provide a better understanding which types of rhythm are present in a dataset. The proximity function can be adjusted depending on the specific question that is asked. Do you want to know if a dataset shows isochrony? Do you want to be able to detect different types of rhythm? What are your expectations? These questions influence which settings you choose. Our study illustrates these differences and options using both theoretical sequences of defined rhythm and 3 different types of animal vocalizations.